Next, what I want to go into is the idea of talking about what happens in the brain when we have Alzheimer's disease. And of course, you might ask the question, why would I want to share any of this stuff with you? Well, hopefully you'll come to the answer that if we can figure out what's going on normally in the system and how it breaks down with Alzheimer's disease, maybe we could then develop interventions to help. So in this next part, we'll talk about the breakdown in Alzheimer's disease and then go into potential treatments for Alzheimer's disease. I've grabbed this picture for you. Hopefully you can see part of it and read the text. Right below it on page 1 is kind of my key terms as well, so hopefully you can type up a story about this. Now, Alzheimer's disease is very complicated in terms of its pathophysiology, but I'm going to try to give you more of a story here that you can understand and you can write out here. It all starts actually with chromosome number 21, which we all have. And it turns out all of us as well have something called amyloid precursor protein. If you look on the bottom of page 1 in the outline, you can see the term. And you can actually see in the picture where it says APP. We all have amyloid precursor protein. It functions in normal cell maintenance and growth for everyone. But it seems when people get Alzheimer's disease, something very important happens to amyloid precursor protein. It seems there are two enzymes that come in and take this long protein, if you will, amyloid precursor protein, and they cut it, they cleave it. And they cleave it, beta-gamma secretases are the ones to cleave it. And when they cleave it, what seems to happen is at the end, there's a peptide left there called beta peptide. And when beta peptide is the leftover fragment, bad things seem to occur. This beta peptide seems to begin to aggregate or cluster in the brain. And when it starts clustering in the brain, it becomes neurotoxic. It spreads this beta peptide into different areas of the brain, and it becomes neurotoxic in terms of plaques taking over in the brain. If you look at the picture, you can kind of see the idea of these toxic plaques occurring in the brain. So that is one part of the process. Amyloid precursor protein cut by beta and gamma secretases forming beta peptide. Beta peptide starts clustering and aggregating in the brain. More and more of it develops, and it leads to this amyloid formation, which is basically, it kills neurons. Big spots of dead neuronal areas in the brain. So this is not good. Now, I need to follow the pipeline, and if you look at the bottom of page 1, you can see I've added a little more to this. A couple of things occur here. First, the inflammation process starts in your brain. The brain goes through the process of inflammation to try to bring in some immune system players and get rid of this plaque. One of those players in the response is called microglia. Micro is small, glia. The microglia comes in and tries through this inflammatory process to get rid of this toxic plaque in the brain. But unfortunately, when people have Alzheimer's disease, this process is ineffective. It does not get rid of the toxic plaque. Instead, it starts killing a lot of healthy tissue and also changes the balance of two important neurotransmitters, acetylcholine and glutamate, in the brain. So this is one big part of the problem that we actually have. That's not shown in the picture here. The other big problem we have is number 2 on the outline here. The other big problem is for people that have Alzheimer's disease, and your book points this out at the end of the Learning and Memory chapter, is a lot of people seem to have a genetic risk factor, if you will. It's called apolipoprotein E4. And it turns out that there are different types of apolipoproteins. There's E2, there's E3, and then there's E4. Now, when apolipoprotein E3 is expressed, what normally happens is when E3 is expressed, you have a neuron, if you will, and a neuron has its shape and structure. When E3 is expressed, basically the neuron gets to keep its shape. If you've ever watched Star Trek: The Next Generation, structural integrity is preserved. The structure is preserved. So when E3 is expressed, the neuron kind of gets to keep its shape. It has its presynaptic region, it has dendrites, axons, cell body, and all that stuff. But it seems for people who have apolipoprotein E4, that doesn't occur. Instead, when E4 is expressed, if you look at the picture, in order to keep your neuron structurally intact, there are tubules. And isn't that a fun word to say? Microtubules. And when E4 is expressed, these microtubules begin to fall apart with this tau protein. And basically what happens is the neuron crumbles. And this leads to the formation of tangles. So for people who have Alzheimer's disease, if they have apolipoprotein E4, this seems to lead to this process occurring where neurons are going to lose their structural integrity and just kind of crumble into nothing, called tangles. So these are the two key problems that happen with Alzheimer's disease. So hopefully you understand with the pictures. You can look stuff up if you wish. And let's see, I think I'll stop this one right here.